Hello, my name is Candace Ward, and in this video we will follow a human as it develops from a zygote to a fetus, focusing on the structural aspects of embryogenesis. We will analyze this organism as it grows from a zygote to a morula to a blastula. We will then examine the process of gastrulation and the development of the primary germ layers before finishing up with an exploration of the process of neurulation. In the upper right-hand corner of your screen, you will notice a blue box marked time, and this will indicate when each of these processes is occurring in our developing organism. In this video, we will analyze the early stages of embryogenesis and associated structures characteristic of each stage. We will explore the embryonic process as the organism begins as a zygote and subsequently develops into a morula, blastula, gastrula, and neurula. By the end of this lesson, you will learn how to answer questions like the one at the bottom of the screen. The MCAT loves to throw a wrench in the works of certain processes and commonly asks you what the resultant pathology would be. The first stage of embryogenesis is fertilization, but let's set the scene. An important fact to remember for the MCAT is that a woman is born with all the eggs she will ever have. Remember from reproduction that these eggs are primary oocytes and are frozen in prophase 1. During menstruation, a single primary oocyte is released from the ovaries. It completes meiosis 1, begins meiosis 2, and then halts at metaphase 2. We now have a secondary oocyte. Fertilization occurs when a haploid sperm cell combines with a secondary oocyte. This combination triggers the secondary oocyte to complete meiosis 2. Another direct result of this fertilization is a cortical reaction, which is indicated by the blue circles around our secondary oocyte and our ovum, and this causes a change in polarity of the cell. This cortical reaction then deposits calcium on the exterior of the ovum, preventing other sperm from fertilizing the same egg. The final stage of fertilization occurs when the male and female pronuclei fuse. A pronucleus is the nucleus of a haploid gamete. So the nucleus from the egg and the nucleus from the sperm cell unite. Now that we have our diploid zygote, let's examine the changes it undergoes. As the zygote moves along the fallopian tubes, it undergoes a series of cell divisions known as cleavages. As soon as the zygote divides, it becomes known as an embryo. Looking at the series of figures at the bottom of the screen, you'll notice that the cell number increases while the total cell volume remains the same. If you look closely at the figure to the bottom left, there's an area marked ZP, and this indicates the zona pellucida, which surrounds the ovum and effectively prevents an increase in volume during very early embryogenesis. When the cell number increases but the volume stays the same, it increases the ratio of surface area to volume, and this gives the cells an increased area for gas and nutrient exchange, which is a common theme you will want to keep in mind for test day. This means that when we have more surface area, we have more room for other processes to occur, such as gas exchange, enzymatic actions, and so on. At this point, these cells have not begun to differentiate. Moving on from the zygote, our next significant embryonic stage is the morula. At this point, we have a solid ball of cells, and the name morula gives us a hint as to the structure at this stage. Morula in Latin means little mulberry, and if you look at the image of the mulberries on the left of the screen and the morula on the right, you'll notice a similarity in structure. So for test day, remember that a morula looks like a mulberry. These cells are located very close to each other, which allows them to communicate very efficiently. Once our organism has reached the morula stage, it undergoes blastulation. And blastulation is the formation of the blastula, which is a sphere of cells surrounding a fluid-filled cavity. And this fluid-filled cavity is known as the blastocele. You can remember the structure of a blastula on test day by thinking about a ball of cells that has had the center blasted out. And on the MCAT, the structure may be referred to either as a blastula or a blastocyst, so it is important to be familiar with both of these terms. The blastula is where we first see differentiated cells. And differentiation means that cells have chosen a path of development and will continue on that path to become different types of cells or tissues. The two differentiated cell types in the blastula are the trophoblasts, which become the chorion and the placenta, and the inner cell mass. The inner cell mass will eventually develop into the entire organism. If you look at our timekeeping box in the upper right corner, you will notice that there are two boxes for the blastula, marked free and implanted. So, up until now, our little organism has been propelled along the fallopian tubes by cilia, cleaving and growing. Then, around day 7, the blastula implants into the thickened uterine lining. And now our organism has access to tons of nutrients from the mother. Now our organism can grow and develop, and its cells can differentiate even further to become tissues like the eye and heart. Over the next couple of weeks, the organism continues to grow implanted in that endometrium. Around week 3, gastrulation occurs, and the inner cell mass of the blastula caves inward to form a tube. This leads to the development of the primary germ layers, which eventually determine what kind of tissue each cell will develop into. The figure on the bottom right will appear several times in this video, and the blue cells will always indicate the ectoderm, the red cells will always indicate the mesoderm, and the yellow cells will always indicate the endoderm. The invagination of the blastula creates a tube made of endodermal cells. This forms the lining of the primitive gut. This primitive gut is known as the archenteron, and the opening of the archenteron is called the blastopore. The archenteron will develop into the digestive tract and associated tissues. Humans are deuterosomes, which means that the blastopore will become the anus. In contrast, the blastopore will develop into the mouth for organisms known as protostomes. Now I'm going to give you a hint for your MCAT as well as future biological studies. If you don't recognize a term on test day or are struggling to learn new terms, you always want to look for word roots. So archenteron has the root entero, which you have seen before in words like gastroenterologist and dysentery. So gastroenterologist and dysentery, what do these two words have in common? They have stuff to do with the intestine, with the gastrointestinal tract. So we know that something is going on in the gut. Entero refers to intestine. Now that prefix arc appears in words like archenemy or archangel and means first or primary. So our archenteron is our first intestine. Now we're moving into a closer examination of each of the primary germ layers. The MCAT loves to test these germ layers, specifically what tissues result from each layer. This may seem a little intimidating at first, having to memorize all the origins of all the different tissues in the body, but don't worry, Kaplan has your back. We have some excellent mnemonics that will make your life a lot easier. Let's start out by looking at our innermost germ layer, the endoderm. This germ layer tends to become the internal organs, so you can remember this layer by recalling that the endoderm forms the end-dermal organs. The endoderm is formed inside the archenteron, which we now know develops into our primitive gut. The archenteron becomes the digestive tract and its offshoots. The stomach and intestines, the main players in our digestive tract, come from the endoderm, as well as accessory organs like the liver and pancreas, which contribute hormones to the digestive process. Our respiratory tract, which is another internal organ, does develop from the endoderm, as well as the bladder. The bladder is actually an outpocketing of the posterior digestive tract, which is indicated by the dashed line, and it will develop in a sex-dependent process. Notable exceptions are the mouth and anus, which are continuous with the outside skin and thus develop from the ectoderm. So this is the main part of the digestive tract that does not develop from the endoderm. Next up is the mesoderm, which we can remember for the MCAT as the mesoderm. A lot of the tissues that develop from the mesoderm involve movement around the body itself moves, and it moves things throughout time, such as genes. When we think about things moving around the body, we should think of the circulatory system, which moves the blood, the excretory system, such as the kidney, which moves waste out of the body through urine, and the lymphatic system, which develops as an outpocketing of the circulatory system, indicated by our little dashed arrow there. And it is important to note that most of the immune system does come from the mesoderm. The musculoskeletal system moves the body itself, and the gonads contribute to the movement of our genes through time, transferring them to the next generation. So we noted that most of the immune system does develop from the mesoderm. However, an exception is the thymus, which develops from the endoderm. Our final germ layer is the ectoderm, which tends to develop into tissues that make a person attractive. Kaplan helps us remember the tissues by calling the ectoderm the attractoderm. So let's explore what this mnemonic device means. You may be attracted to another person because of their appearance or personality. Tissues like the skin and hair develop from the ectoderm, as do the teeth and eyes. If you like a person's dark hair or their straight teeth, you can thank their ectoderm. In terms of personality, a person's nervous system develops from the ectoderm, so their brain and peripheral nervous system come from the ectoderm. So the fact that someone can speak and hear what you say is an attractive quality which we can now remember comes from the ectoderm. While most of our sensory structures come from the ectoderm, a notable exception is the taste buds, which develops from the endoderm. The final process in early embryogenesis is neurulation, which is the formation of the early nerve cord. And neurulation begins with the formation of the notochord by mesodermal cells. If you look at the figure at the bottom of the screen, you will note that the notochord is located at the top. This is actually the back of our organism, which is called the dorsal side. The notochord is located beneath the ectodermal neural plate. These mesodermal cells in the notochord induce the ectodermal cells in the neural plate to contort into neural folds, which surround a neural groove. And the tips of these folds are neural crest cells, which will eventually differentiate into further tissues. After the neural folds fuse, the neural tube is formed and subsequently separates from the surface. The neural crest cells migrate and become the peripheral nervous system, pigment-producing cells, and some glia. Let's try a couple of practice questions to test our knowledge. This question asks about the number of cytokinetic steps since fertilization. Take a second and pick an answer. A common wrong answer is 4 times, since 2 to the 4th is 16, but that's not an answer choice, so we know that something is up. So let's think. What occurs after fertilization? When that sperm cell first burrows into the secondary oocyte, it triggers the egg to complete meiosis II, which ends with cytokinesis. So we have 1 more cytokinetic step to the 4 further divisions it takes to yield 16 cells. And our answer is B, 5. Let's try another question. This question asks us to analyze what would happen if we couldn't cause those first mesodermal cells to differentiate. So I want you to think about what role the mesodermal cells play in development. The first question we need to ask ourselves is when we saw those first mesodermal cells appear, which was during gastrulation. Next, let's think about what role mesodermal cells play in further division. When we explored neurulation, we discovered that mesodermal cells in the notochord trigger the formation of the neural tube. And if those initial mesodermal cells couldn't become the notochord, neurulation can't occur and the organism will be stuck as a gastrula, which is answer choice B. We explored the 5 main stages of early embryogenesis: the zygote, the morula, the blastula, the gastrula, and the neurula. The zygote was formed from the union of a haploid sperm cell and a haploid secondary oocyte. Once the cell began to divide, it became known as an embryo. We reached the morula stage once about 16 to 20 cells were formed. The morula looks like a mulberry, so its cells are in tight communication with each other. The blastula looks like a solid ball of cells with its center blasted out. After it travels along the fallopian tubes, it reaches the uterus and implants in the thickened endometrium. This is so it can receive nutrients from the mother and further develop. Gastrulation occurred about 3 weeks in, and the blastula indented to form the primitive gut. The germ layers develop at this stage, and the MCAT loves to test those germ layers, so remember those Kaplan mnemonics. Finally, with neurulation, we develop the early nerve cord, which will eventually become the spinal cord. We went pretty in-depth about the early stages of embryogenesis in this video, but with further study, you'll be able to master these concepts and successfully apply them on test day. Thank you.